Hello people. Blood is actually regarded as a liquid connective tissue in which cells are floating around and they are not stationary. If you, for example, take a sample of blood and put it into a tube with an anticoagulant and take it to a centrifuge device, you will get two distinctive parts of blood. One of them is blood plasma, and the other is actually the cellular part of blood. This part is representing the cellular part. A normal average human has about 5 liters of blood in its system, and about 55% is blood plasma, and about 45% are cells. 1% or less is also a cellular part of blood. Plasma, if we take a look at the structure of plasma, is mostly water. About 92% of plasma is water, and it is also made of proteins. About 7%. So together they are 99% of the structure of plasma. There is 1% left. Even though this 1% is small, it is very important because it represents ions, nutrients, hormones, gases, and everything else that is not here. So this is basically the structure of plasma. Proteins can be further subdivided into albumins, which are about 60% of all proteins in plasma. And albumins are actually proteins that are very important for regulating blood pressure. They are also globulins, like immunoglobulins, and there are also fibrinogens, which are big proteins that play a big role in blood clotting. When it comes to the cellular part, the important part is these 45%. They are representing the hematocrit or the erythrocytes. This 1%, it is actually representing leukocytes plus thrombocytes. In an average human, in a cubic millimeter of blood, there are 5 million erythrocytes. About 5 million erythrocytes in a cubic millimeter of blood. There are 4,000 to 9,000 leukocytes in a cubic millimeter of blood, and there are about 150,000 to 400,000 thrombocytes in a cubic millimeter of blood. So, if we now take a look at the erythrocyte, erythrocytes are cells that have the important function of gas transport. And in order to maintain this function and to function in the bloodstream, there are two key points to understand about the erythrocytes. First is their shape, and secondly, it's the cytoskelet. So the shape of the erythrocyte, as we can see here, it's biconcave shape. That means that in the central part, the erythrocytes are caved in. This is important because this shape allows the erythrocyte to have a 20 to 30% bigger surface area than if it was a spherocyte. The second important thing about the erythrocyte is its cytoskelet. The cytoskelet of the erythrocytes is the one thing keeping its shape and allowing it to be deformable, having this function of deformability. The cytoskelet is made up of many proteins. Some of these proteins, there are many, but the commonly mentioned are ankyrin proteins, spectrin, proteins, actin proteins. There are also myosin 2 proteins and other proteins like band 3 proteins. And all of them together function in order to make this cytoskeleton. Band 3 proteins are proteins that are in the membrane of the erythrocyte. They are transmembranous proteins. On the inside are ankyrin proteins enabling the bind to actin spectrin part proteins. And together they make this cytoskelet or skeletal web inside the cytoplasm. The erythrocyte has a diameter of 7.5 micrometers, and the smallest capillaries have a diameter of 5 to 10 micrometers. This gives actually the rise to a problem that erythrocytes need to pass through blood vessels that are smaller in diameter than they are themselves. And in order to do this, they have this function of deformability, so they can actually deform from this shape to a deformed shape in order to pass through these capillaries. And they would be destroyed if it wasn't for this cytoskeleton. All these two functions are so very important for the erythrocyte to pass through blood vessels, to travel through the bloodstream, and to have this function of gas transport that is enabled through hemoglobin. Hemoglobin is actually making up 1/3 of the erythrocyte. Hemoglobin can bind gases like oxygen, becoming oxyhemoglobin, carbon dioxide becoming carbaminohemoglobin. It can also bind other gases like nitrogen oxide and carbon monoxide. The erythrocyte is actually made up 50% of proteins. It has also its own enzymes and that are producing a little bit of energy through anaerobic use of glucoses, and it also keeps these proteins functional. These enzymes don't allow their oxidation. In about 120 days, these enzyme supplies are depleted, and the erythrocytes proteins start to degrade, and then they are eliminated by the macrophages in the spleen and in the liver. The erythrocyte is being produced in the bone marrow. The second important function is that erythrocytes, if they wouldn't have this shape, they would be eliminated. So there are actually diseases in which gene mutations make then faulty proteins or proteins that don't have this physiological structure, like hereditary spherocytosis. In this disease, the erythrocyte actually has a spheric shape because a mutation in spectrin. And these spheric erythrocytes, or spherocytes as they are called, they can still do gas transfer. If they couldn't, the patient would immediately die. But the problem is they are being fastly eliminated in the spleen and in small capillaries because they are prone to physical damage, because their cytoskeleton isn't able to maintain their membrane. In order to, for example, treat this disease, you can remove the spleen or do a splenectomy, so the anemia will be a little bit better. Thank you.